Hi everyone, I am Anmol CK from first MSc Biology St. Mary's College. Today we are discussing about the topic gene therapy. Gene therapy is the introduction of genes into existing cells to prevent or cure a wide range of diseases. It is a technique for correcting defective genes responsible for disease development. The first approved gene therapy experiment occurred on September 14, 1919 in the US when Ashanti D. Silva was treated for adenosine deaminase SCID. Two major gene therapy. In gene augmentation therapy, a DNA is inserted into the genome to replace the missing gene product. In case of gene inhibition therapy, the antisense gene inhibit the expression of the dominant gene. This is a picture of two major gene therapy, gene augmentation therapy and gene inhibition therapy. Approaches for gene therapy. There are two approaches for gene therapy. First one is somatic gene therapy. The non-reproductive, that means non-sex, cells of an organism are referred to as somatic cells. These are the cells of an organism other than sperm or egg cells, example bone marrow cells, blood cells, skin cells. At present, all the research on gene therapy is directed to correct the genetic defect in somatic cells. In essence, somatic cell gene therapy involves the insertion of a fully functional and expressible gene into a target somatic cell to correct a genetic disease permanently. Germ cell gene therapy. The reproductive sex cells of an organism constitute germ cell line. Gene therapy involves the introduction of DNA into germ cells is passed on to the successive generation. For safety, ethical and technical reasons, germ cell gene therapy is not being attempted at present. The genetic alterations in somatic cell are not carried to the next generations. Therefore, somatic cell gene therapy is preferred and extensively studied with an ultimate objective of correcting human diseases. There are two types of gene therapy. First one is ex vivo gene therapy. This involves the transfer of genes in cultured cells, example bone marrow cells, which are then reintroduced into the patient. Next, in vivo gene therapy. The direct delivery of genes into the cells of a particular tissue is referred to as in vivo gene therapy. Ex vivo gene therapy. The ex vivo gene therapy can be applied to only selected tissues whose cells can be cultured in the laboratory. The technique of ex vivo gene therapy involves the following steps. Isolate cells with genetic defect from a patient, grow the cells in culture, introduce the therapeutic gene to correct gene defect, select the genetically corrected cells and grow, transplant the modified cell to the patient. The procedure basically involves the use of the patient's own cells for culture and genetic correction, and then they are returned back to the patient. Ex vivo gene therapy is efficient only if the therapeutic gene is stably incorporated and continuously expressed. This can be achieved by use of vectors. Vectors in gene therapy. The carrier particles or molecules used to deliver genes to somatic cells are referred to as vectors. The important vector employed in ex vivo gene therapy are listed below. Viruses, human artificial chromosome, bone marrow cells. Viruses. The vectors frequently used in gene therapy are viruses, particularly retroviruses. RNA is the genetic material in retroviruses. As the retroviruses enters the host cell, it synthesizes DNA from RNA. By reverse transcription, the so formed viral DNA gets incorporated into the DNA of the host cell. The proviruses are normally harmless. However, there is a tremendous risk since some of the retroviruses can convert normal cells into cancerous ones. Therefore, it is absolutely essential to ensure that such a thing does not happen. Human artificial chromosome HAC. The HAC is a synthetic chromosome that can replicate with other chromosomes besides encoding the human protein. As already discussed above, the use of retroviruses as vectors in gene therapy is associated with a heavy risk. This problem can be overcome if human artificial chromosome is used. Some success has been achieved in this direction. Example for ex vivo gene therapy. Therapy for adenosine deamine deficiency. The first and the most published human gene therapy was carried out to correct the deficiency of the enzyme adenosine deaminase. This was done on September 14, 1919 by a team of workers led by Bliss and Anderson at the National Institute of Health, USA. Girl's name Ashanti, 4-year-old. In vivo gene therapy. The direct delivery of the therapeutic gene into the target cells of a particular tissue of a patient constitute in vivo gene therapy. Many tissues are potential candidates for this approach. These include liver, muscle, skin, spleen, lung, brain, and blood cells. Gene delivery can be carried out by viral or non-viral vector system. The success in vivo gene therapy mostly depend on the following parameters: intracellular degradation of the gene and its uptake by nucleus, the efficiency of the uptake of the remedial gene by target cells, the expression capability of the gene. Gene delivery by viruses. Many viral vector systems have been developed for gene delivery. These include retroviruses, adenoviruses, and herpes simplex virus. Gene delivery by non-viral systems. There are certain limitations in using viral vectors in gene therapy. In addition to the prohibitive cost of maintaining the viruses, the viral proteins often induce inflammatory responses in the host. Therefore, there is a continuous search by researchers to find alternatives to viral vector system. The non-viral systems are listed: pure DNA construct that can directly introduce into a target tissues, lipoplexes, lipid DNA complexes that have DNA surrounded by lipid layers, human artificial chromosome which can carry large DNA. Gene therapy strategy for cancer. Cancer is the leading cause of death throughout the world, despite the intensive treatment strategies: surgery, chemotherapy, radiation. Gene therapy is the latest and a new approach for cancer treatment. Some of the development are briefly described. Tumor necrosis factor gene therapy. Tumor necrosis factor is a protein produced by human macrophage. Tumor necrosis factor provides defense against cancer cells. This is brought by enhancing the cancer fighting ability of tumor infiltrating lymphocyte, a special type of immune cells. The tumor infiltrating lymphocyte were transformed with a tumor necrosis factor gene and used for the treatment of malignant melanoma, a cancer of melanin producing cells usually occurs in skin. Then TNF as such as highly toxic and fortunately no toxic side effects were detected in the melanoma patients injected with genetically altered TILS with TNF gene. Some improvement in the cancer patients was observed. Suicide gene therapy. The gene encoding the enzyme thymidine kinase is often referred to as suicide gene and is used for the treatment of cancer. Thymidine kinase phosphorylate nucleoside to form nucleotides which are used for the synthesis of DNA during cell division. The drug ganciclovir bears a close structural resemblance to certain nucleosides. By mistake, TK phosphorylate ganciclovir to form triphosphate GCV, a false and unsuitable nucleotide for DNA synthesis. Triphosphate GCV inhibit DNA polymerase. The result is that elongation of the DNA molecule abruptly stops at a point containing the false nucleotide ganciclovir. Further, the triphosphate ganciclovir can enter and kill the neighboring cancer cells, a phenomenon referred to as bystander effect. The ultimate result is that cancer cells cannot multiply and therefore die. Thus, the drug ganciclovir can be used to kill the cancer cells. Ganciclovir is frequently referred to as prodrug, and this type of approach is called prodrug activation gene therapy. Ganciclovir has been used for treatment of brain tumors. This is a figure the action of ganciclovir mediated by thymidine kinase to inhibit the growth of cancer cells. Antigen and antisense therapy. In general, gene therapy is carried out by introducing a therapeutic gene to produce the defective or the lacking protein. But there are certain disorders which result in the overproduction of certain normal proteins. It is possible to treat these diseases by blocking transcription using a single-stranded nucleotide sequence that hybridizes with a specific gene, and this is called antigen therapy. Antisense therapy refers to the inhibition of transcription by using a single-stranded nucleotide. Further, it is also possible to inhibit both transcription factors responsible for the specific gene expression. Nucleic acid therapy refers to the use of DNA or RNA molecules for therapeutic purposes as stated above. The naturally occurring sequences of DNA and RNA or the synthetic ones can be employed in nucleic acid therapy. Antisense therapy for cancer. Antisense RNA molecules are frequently used in cancer therapy. This approach is effective only if the antisense oligonucleotide specifically bind to the target mRNA and block protein biosynthesis. This can be achieved in two ways. This antisense cDNA can be cloned and transmitted into the cells. Antisense mRNA is synthesized by transcription. This can be readily bind with a specific mRNA and block translation. The other way to block translation is to directly introduce antisense RNA into the cells. This hybridizes with the target mRNA and block translation. The antisense mRNA therapy was tried for the treatment of brain tumor, namely malignant glioma and the cancer of prostate gland. In case of malignant glioma, the protein insulin-like growth factor 1 is overproduced, while prostate cancer insulin-like growth factor 1 receptor protein is more synthesized. For both these cancers, the respective antisense mRNA molecule, these in turn are used to block translation. Peptide nucleic acid therapy. PNAs, that is peptide nucleic acid therapies, are artificial analog of nucleic acid with a polypeptide backbone. They possess standard nucleic acid bases attached to a polypeptide in place of sugar phosphate backbone. And since PNAs have been developed to inhibit translation of HIV viral transcript gap pol and to block translation of cancer genes. Example HAS-RAS and BCL2. Conclusion. Theoretically, gene therapy is the permanent solution for the genetic diseases, but it has several complexities. As its current stage, it is not accessible to most people due to its huge cost. A breakthrough may come any time, and a day may come when almost all diseases will have a gene therapy. Reference. Thank you.